Ladies and gentlemen, my name is Mariya Krestina, and the topic of my work, 1H NMR spectroscopy after alcohol impact of red tissue and blood. Okay, first of all, I would like to start with definitions of some words: metabolomics, and metabolome. Metabolomics studies the final and intermediate products of metabolism. Metabolomics studies the metabolic response to pathophysiological impact or genetic modification. It studies the metabolic changes. Metabolome is a complete set of metabolites which can be found in the biological sample. Metabolome has a very important feature. It is unique like human fingerprint. So it is very great biological characteristics. Here you can see the number of metabolomics and metabonomics paper. As you can see, this field is growing and development. And why a lot of scientists are so interested in this field? I think that the reason for it is hidden in definition of metabolism. Metabolism is a set of chemical reaction which occurs in a living system for maintaining a life. So, metabolism reflects the state of whole organism. Metabolic changes are the earliest response, and metabolism has a unique feature. So, every pathology or treatment first of all leads to metabolic changes. As a result, metabolic changes can be served like a pathology marker or index of treatment efficiency. There are two most important methods for metabolomic studies: NMR spectroscopy and mass spectrometry. Both of them has pluses and minuses, of course. NMR spectroscopy requires special sample preparation, but it is possible to determine concentration of all detectable metabolites during one measurement. One calibrant is enough, and in vivo and in vitro studies are available. Mass spectrometry also requires special sample preparation, sample separation, calibration curve is necessary, and just in vitro studies are available. These properties of NMR spectroscopy made it indispensable for biological studies. But in fact, of course, NMR spectroscopy and mass spectrometry complement and confirm each other very well. The aim of my work is the investigation of alcohol exposure of laboratory rats with help of 1H NMR spectroscopy. We solved following task: to study brain, liver, and blood metabolism after alcohol impact, to study dynamics of blood metabolism, and to verify the opportunity of usage blood metabolism for monitoring the liver state. Few words about experimental pathology model. We divided 40 laboratory rats into 4 groups. The first group was given water, the second water and medicine, the third alcohol, and the fourth alcohol plus medicine. Medicine was given nitrogastrically once a day, and 50% ethanol solution was used like alcohol, and it was given also intragastrically but twice a day. Duration of pathology model was 28 days. Every daily dose of alcohol was 10-12 grams per 1 kilogram of animal weight, and dose of medicine is 0.1 gram. Like medicine, we use extract of mushroom reishi, Ganoderma lucidum. It's here. As I said earlier, NMR spectroscopy and mass spectrometry require sample preparation. After literature analysis and approbation some methodics, we choose the following sample preparation protocols. At first, tissue is homogenized in ethanol solution. After that, the sample is ultrasonicated and centrifugated, and tissue parts are removed. Then, in order to separate hydrophobic and hydrophilic metabolites, we edited chloroform and water in proportion 1:1, and then it's again centrifugated and hydrophobic metabolites are removed. This stage repeat one more time, And finally, the sample was dehydrated by vacuum drying, and fluid are completely removed. Here you can see just a representative NMR spectrum, just one part, because later also exist. Blood, brain, and liver. We made the spectra for all animals, for all tissues, and for blood. The next step is the identification of spectra. It's a very complicated task. It is necessary to understand which lines correspond to which metabolites. So at first we use mass spectrometry, method IC cutoff, with help database HMDB and METLIN, and isotope analysis we found some set of metabolites. But of course not all of them can be detected in liver, brain, blood, so we checked it with other database, and after that we found other set of metabolites, and this set of metabolites we compared with our experimental data and found, and we can detect it and quantify just 43 liver metabolites, 38 brain metabolites, and 27 blood metabolites in vitro, and 7 of brain in vivo. Tissue spectrum require calibration. We normalize all tissue spectra because it is really impossible to take exactly the same amount of tissue, and the tissue is not homogeneous. So here you can see it's just the sum of all detectable metabolites, and it's don't have reliable changes. But blood sample preparation is allowed to take exactly the same volume of blood, and maybe it can reflect more deep effects. Like this, we can see that alcohol and alcohol plus medicine are some of all detectable metabolites decreased. So it's the test and reliable changes. Some words about liver metabolism, just some few interesting maybe. It's a proliferation process. We found that amino acids concentration decreased, and it leads often to protein synthesis. Protein synthesis leads to formation of new cells. It was found as a result of histology analysis that amount of binuclear hepatocyte were increased. So it also leads to formation of new cells. And we can conclude that the first reaction of liver is just an increasing of the amount, the volume, proliferation process. Few words about brain metabolism, about neurotransmitter. It is known that ethanol leads to inhibition of nerve pulses. And we found that such neurotransmitter like glutamate and aspartate, their concentration is increased in brain. So it is a reaction of brain, because it's a stimulating neurotransmitter, to try increase nerve impulses. And also ethanol, according to the literature, leads to depression. And according to other articles, increasing amount of this neurotransmitter also leads to depression. Minuses of this is just stimulating neurotransmitter promotes the penetration of calcium 2 plus inside of cell. And we found that taurine is the other neurotransmitter, their amount increased in ethanol group. And according to the literature, increasing of taurine also leads to it's inhibition of penetration calcium 2 plus inside of cells. So taurine defends and protect cells from toxic reaction due to calcium 2 plus. As Yes. We take blood in the middle and in the end of experiment. We made two control points. To use blood analysis is very convenient and more easier for clinical researchers and for metabolomics also, because we can monitor the development of treatment or disease. We found that after 2 weeks, just 6 metabolites have reliable changes. And after 4 weeks, 12 metabolites have reliable changes. So, of course, ethanol impact is increasing during the time. Here I would like to tell you about method PLS-DA, just about what this plot. This method allows to represent a special separation between experimental group. This axis is a linear combination of metabolites, and these points are animals. So here just also every color represent experimental group, and just the average mean and square error. So here you also can see that after 2 weeks and after 4 weeks, the special separation is better. So alcohol impact is increasing. And we found this method was performed also for brain, blood, and liver. And here you can see that it's a very similar behavior. Just a group which were given ethanol, they are suited in one side of plot, and just the water with other side. About blood. Of course, blood is very convenient, but could blood reflect correctly the liver metabolism? We calculated correlation coefficient and found that 5 metabolites correlate between blood and the liver. And 4 of them have similar reliable changes. Here you can see that the changes are similar. And this is just a picture about small comparison in vivo and in vitro spectroscopy. This in vitro spectrum, this also in vitro, but with exponential adiposization, and here in vivo. So they are quite similar. And conclusion. Liver, brain, and blood metabolism were considered after alcohol impact. Dynamics of blood metabolism correctly reflect the state of liver, and it is possible to monitor liver health with blood spectroscopy, but of course not all blood metabolites reflect liver state. So, thank you for your attention.
Thank you. Here's your questions to Mariya. Oh, yes, first.
Does the in vivo studies means that you put in ampoule in spectrometer full cell, living cell?
Not of course, it was a rats.
Rats and...
Rats. When we made modeling of pathology, we made tomography and in vivo spectroscopy also. After that, we removed tissue and frozen that and go to the preparation for in vitro studies.
Yeah.
But before it was in vivo spectroscopy with rats in biospec 11 Tesla.
Thank you.
And 2 weeks, 4 weeks, the same animals? Or
Yes, of course, the same animals.
Same.
Yes, it was dynamics, the same animals.
In the singles? yes?
Yes.
Many examples, but the analysis from the single during 2 weeks, 4 weeks, and so on, yes?
Не совсем понимаю. I'm sorry.
As usual, we use many examples for investigation,
Yes.
but we can analyze one group after 2 weeks, another group after 4 weeks.
Oh, no. We analyze 4 weeks after 2 weeks. 4 groups after 2 weeks and 4 groups after 4 weeks. So 40 animals every time.
Okay.
What's the control group?
Just the water. Yes. The first group was given a water. It was a
group of animals.
yes. This group was given just water because it was necessary to take similar stress conditions. So we just intragastrically given the water.
Please.
И все-таки как вы оцениваете относительную роль в этих делах НМР и масс-спектрометрии? Вы использовали довольно дорогостоящее МР-оборудование, 605 микроцерой.
Да.
Спектрометр, а, наверное, масс-спектрометр был у вас какой-нибудь?
У нас был-
Простенький.
Нет, масс-спектрометр у нас тоже был хороший. Я не пишу про него, потому что... Нет, мы делали осикутов, то есть это электроспрей, и это не электронная ионизация, это...
Выводы обоих методов совпадали?
Мы не оценивали количественные результаты по масс-спектрометрии, так как это гораздо сложнее делать. И тем более мы хотим дальше сравнивать с инвиво данными. Поэтому нам необходим ЕМР для того, чтобы как раз вот проводить то, что я здесь показываю. То есть вот здесь можно как раз проинтегрировать вот это и сделать выводы, может ли инвиво спектроскопия корректно оценивать, когда мы знаем, что инвитро, она точная.
Вы как-то ничего не сказали об окрестностях, там группа были же, да?
Да, но я каждый... Вот, у меня нарисованы усы везде. Вы видите? Вот эти черненькие палочки. Вот. Ну и помимо этого мы использовали статистический анализ, множественное сравнение средних, которое показывает, отличаются ли достоверно группы между собой. То есть нельзя просто так по усам даже сказать. То есть нужно составить статистическую гипотезу и проверить ее. И вот я здесь пишу как раз, когда А и Б это разные группы, то есть они достоверно отличаются друг от друга.
Еще вопрос.
How can we see only brain or only liver in in vivo?
We see just the brain. I don't spoke about liver in vivo. I show you just brain. It's a brain.
Yes, brain or blood in live animals.
Yeah, but blood you can take a blood analysis. You cut хвост. I don't know how to say it. And yes, other it is possible. Yes, I just saw it, but I don't cut.
Этот спектр ЕМРовский можно пояснить, как вы его получали, да? Если получали.
Какой?
Вот нижний.
Нет, нижний я не получала, я его только обрабатывала. Я получала всю инвитро спектроскопию.
Вы не знаете условия получения этого спектра, да?
Ну, я вам примерно... А что вас интересует? Я присутствовала просто при экспериментах.
На каком приборе и...
Прибор?
Помещалось внутрь вот этого поля, да?
Да.
Прекрасно маленький трубик, да, и копилось, да? Просто я смущен, что очень большой шум. На 500 мегагерц можно было его вытянуть с потолка и выше. Почему такой зашумленный? Я просто этим удивлен.
Ну, я думаю, что просто я его хорошо не отгладила.
Нет, почему? Это же как бы функциональная томография. Там чувствительность какая?
Да, спектроскопия в окселе.
Маленький, миллиметр на миллиметр.
Да. Ну, мышка помещалась в прибор, да, 500 МГц, была под наркозом. И...
Удивляюсь, буквально 10 лет назад подобные спектры, где отношение нашел 100 к 1, там, 200 к 1, а у вас он как бы в шумах.
Это кажется заполнение.
Нет, и тоже...
Тоже мышка, да, ну там копили
да?
часов 5,
Ну да, но здесь всего 10 минут.
копили часов 5.
А здесь
Здесь 10 минут. Где-то это 10 минут.
Ну, наверное, поэтому, да. Ну тогда малоинформативный. Тогда какая же концентрация определена, когда тут такой
Я концентрацию определяла по инвитро спектрам, по вот этим красивеньким. То есть инвиво здесь в качестве, я просто показываю, что дальше возможно перейти от инвитро спектроскопии к инвиво, и что действительно спектры хорошо отражают друг друга.
Okay, my question about the role of medicine, which you extracted from this mushroom. Are you expected, and what is the result? Is this medicine for treatment or another additional impact plus alcohol?
Yes, it's not a treatment. Thank you for your question. We just Unfortunately, as you can see here, for example, that medicine is made better, but here it's not reliable changes between medicine and alcohol. And in fact, about 2 animals were died because of this medicine. We make a surgical we just see on the stomach, and stomach was very поврежден. I say it. Thank you.
Один вопрос я могу задать только по-русски. А у вас не было такого эксперимента, чтобы еще вместо лекарства что-нибудь для похмелки?
Нет,
Не было?
такого не было.
Так, чтобы для нас польза какая. Excuse me.
Who knows?
Just well. Please, any questions? Yes.
Would you please show me the slide about the brain chemistry?
I was here.
Yeah, this one. I don't know what your resources are, but my experience tells me that the more ethanol there is, the less depression, not the other way around.
I just read about animal depression, and it was not chronic. Chronic consumption leads to depression. Yes, chronic consumption, of course, it was 1 month.
Okay, stimulating neurotransmitters means more dopamine and more serotonin. That means more happiness.
Yes, but also glutamate is present in brain, and it is a neurotransmitter, and I read about glutamate and aspartate literature, not about this conclusion, not from stimulating neurotransmitter, just from glutamate and aspartate.
Okay, thank you.
Thank you.
I should stop this discussion. Thank you very much for your.
Thank you.